Enrichment only comes through purification. This is a yoga quote, but it's also applicable to proteins, in particular enzymes. As you purify an enzyme, you enrich your sample with your protein of interest, and in order to study and characterize a protein, we really do need a pure source for your experiments. By the end of this lecture, you should be able to explain how proteins can be separated based on differences in their size, charge, affinity, and hydrophobicity. We'll see that there's lots of different methodologies that can be used to purify a protein from a complex mixture. And then once you think you've purified a sample, you can actually use an SDS page gel to really assess whether or not your sample is pure. Now, most proteins are colorless, so we will look at various ways of visualizing and quantifying a protein in solution. These are the supplemental readings also posted on Quarkus. Proteins in the cell definitely do not practice physical distancing. It's a very crowded environment, and in order to characterize a protein, we need to be able to separate it from all the other molecules that are found in the cell. The reason why we need to separate it from these molecules is because they might interfere with your experiments or even modify your protein, creating a heterogeneous population. And this would make it very hard to study and control for variability. Now, we've already looked at the basics of protein structure, and it's these differences in properties that can be exploited in order to purify them. The very first thing you need to do is choose the right cell or tissue source in order to obtain an abundant quantity of protein. So fibrin and albumin were some of the first proteins that were studied in the early 1800s, simply because you could isolate them from blood. You can then lyse or break apart the cells in order to purify the intracellular proteins, and this will generate what we call a crude extract. By spinning down your sample at very high speeds using a centrifuge, you can precipitate out larger organelles or molecules that might not be soluble under different conditions. But just remember that some proteins may not necessarily be soluble and may be found in your pellet, but centrifugation is one way you can start to purify your protein. Now, if you can't find a good source or have trouble purifying the endogenous source of protein, molecular biology is typically used in the lab in order to genetically engineer your protein of interest. And we'll see some examples of a tag that you could use to help with this process. There are a lot of ways of breaking open a cell, including more physical methods like grinding or sonication using very high frequency, as well as vortexing with glass beads. But you can also use osmotic pressure where water can flow into the cell and break them apart. Detergents are amphipathic molecules that we'll look at in more detail in the membrane module, but they can also be used to break apart the noncovalent interactions holding the cell membrane together. Just keep in mind that when you do lyse a cell, you may also release proteases or enzymes that can chew up your proteins of interest, so that's why many purification protocols will be done in the cold room to lower the activity of these enzymes. In addition to heat playing a role in enzymatic activity, it can also promote the unfolding of your protein, which we call denaturation, since it's mostly these non-covalent interactions that are holding your protein together. So this is another reason why samples are generally kept on ice. The pH of the buffer will also be tightly controlled, as we learned that many functional groups can be protonated or deprotonated, and this obviously can play a role in your protein's structure and function. There are four main types of chromatography that separate your protein based on different properties. Remember from chemistry that chromatography is a technique used to partition molecules between a mobile phase, like a buffer, and a stationary phase, which in biochemistry is usually some sort of column. We can separate proteins based on either their size or shape using size exclusion chromatography, and this technique also goes by the name of gel filtration. If we separate proteins based on their charge, this would be ion exchange chromatography, and their ability to bind to special molecules or ligands is known as affinity chromatography. The last property is hydrophobicity, and this type of chromatography is known as reverse phase high pressure liquid chromatography, or HPLC. If you're interested in gaining lab experience, our BCH 370 lab course allows you to actually purify some samples of protein using different techniques. Size exclusion chromatography uses a column that, as the name suggests, has beads that exclude molecules of a certain size, and by size we're talking about the molecular weight of the entire protein, including multiple subunits if they are held together by the various interactions. This results in smaller molecules traveling slower through the column, such as these proteins represented in pink, while proteins that are above the molecular weight cutoff, like these yellow ones, go straight through and come off earlier. Anything in between the range of the column will spend an intermediate time on the column, partitioning between the buffer and the stationary beads. Now, if a molecule has a more elongated structure, it might tumble through solution and actually appear larger than its actual molecular weight, which, remember, is based on the atoms that are present. For these types of columns, you would run standard proteins of known molecular weights separately so that you could have an idea of when or in which fraction your protein will come through the column. The fraction where your protein comes off of the column is also known as the elution volume if you're measuring the volume of liquid that is coming off of the column. Now, the range of molecular weights where you can separate one protein from another is usually called the fractionation range, and you can buy different columns with different fractionation ranges if you have an idea of how big your protein actually is. One concept we need to review is absorbance, and it's one way we see proteins coming off of a column. Now, most proteins don't absorb light in the visible range, and they're not pink or yellow or blue like I've represented them in the last slide. They're typically colorless, but do absorb light differentially at different wavelengths. Now, this is due to the presence of different bonds, and in particular the aromatic amino acids, where tryptophan is actually one amino acid that can absorb the most light at 280 nanometers. Each protein will have different amounts of tryptophan as well as tyrosine and phenylalanine, and tyrosine can contribute a bit to the absorbance at 280, so we can use this property to figure out how much protein is coming off of the column. You can also use stains like Coomassie blue to permanently stain and detect your protein coming off the column, and there are experiments like a Bradford assay that'll quantify how much protein is present or the concentration of total protein. We also need to make sure that we have enough protein to work with, and the aromatic amino acids allow us to monitor how much protein is present based on their ability to absorb light. But keep in mind that if you do still have a mixture of proteins, it can only tell you the total amount of protein present, not if one protein is present versus another. Hopefully you remember the Beer-Lambert law from chemistry, where if you shine light on a sample, you can use a filter to select for a specific wavelength, and for proteins it would be 280 nanometers, and we can then measure the difference between how much light is transmitted and the incident beam. This will measure the absorbance of your sample, and this is related to the log of the incident beam over the transmitted light. The absorbance is also a function of the molar extinction coefficient, which depends on the number of aromatic amino acids, the concentration of protein in your sample, and the length of the cuvette holding your protein sample. So we can measure the absorbance and use this relationship to figure out the concentration of protein in your sample. Now, just to come back to our size exclusion column, when we measure what is coming off the column, you'll usually see data presented like this in an elution profile, where the volume of the buffer coming off of the column is plotted on the x-axis, and the concentration of protein is on the y-axis. When molecules go straight through the column, this is called the void volume, representing anything that is larger than the size of the pores and is not retained on the column. The elution volume would be where a molecule comes off the column, and this could be the void volume if it doesn't partition in the beads. The total volume of the column is the lowest range of fractionation where the smallest molecules spend the most time on the column. When you are picking a column, you would look for a size of beads that would separate and elute your protein of interest between V0 and VT. And you can see that the elution of molecules may happen over a range of volumes. It's the area under this curve that would be the total amount of protein coming off of that column at any given point. The second main type of chromatography used is to use a stationary phase that has a charge on it, so that proteins with the opposite charge will bind and are separated from the mixture. Now, remember we talked about the pi of an amino acid or a polypeptide? You can also calculate it for very long polypeptides or complete proteins, and this can tell you what the charge of a protein is at any given pH if you are either above or below that isoelectric point. So when the pH is below the pi, functional groups will be protonated, so your protein will be positively charged. In this cartoon, we see that a positively charged protein will bind to a negatively charged bead. And these beads or resin are called cation exchange resins since they are binding to cations or positively charged proteins. Any proteins that are neutral or of the same charge will go straight through the column. Carboxymethyl is a common molecule that can act as a cation exchanger when it's bound to the bead that's either consisting of cellulose or agarose solid supports. On the flip side, anion exchange resins like DEAE will attract and bind to negatively charged peptides due to the positive charge on the DEAE resin. After you've washed off anything that doesn't bind, so anything that's neutral or the same charge of the column, you can then elute or unbind your protein by either changing the pH so that your functional groups become protonated or deprotonated, or even add a buffer that's equal to the pi of your protein so that the net charge becomes 0 and your protein comes off the column. Another way you can elute proteins from an ion exchange column is to increase the salt concentration. So this will compete for binding to these electrostatic interactions. Now, part of your protein structure assignment will involve identifying the pi of your protein and explaining how the protein was purified to determine its structure. Affinity chromatography also works on the basis of attraction, where your column will contain a molecule or ligand that your protein likes to bind to. For instance, a glucose binding protein, shown here in yellow, could interact with a solid support that has a covalently attached glucose residue, represented by the letter G. Once you've washed and removed any proteins that don't have this affinity for the column, you could then use free ligand to compete for binding and elute your protein of interest. Now, this technique is also a great way to concentrate your protein and only have it come off the column in a very small fraction of buffer. Another example of affinity chromatography you may have heard about is the binding of His tags to a nickel NTA resin. You might use this technique if you're having trouble purifying your protein using the other types of chromatography, or if you aren't able to find a good tissue source for your protein of interest. We can use molecular biology to add a His tag consisting of 6 to 10 histidine residues in a row onto your protein of interest, and we usually add it to either the start or end of the protein so it doesn't interfere with its structure. By including it at the ends, so either at the N terminus or the C terminus, this may also allow for it to be accessible and hopefully not affect the function of the protein. The experiment is just shown on the right, where you would express your tagged protein in a cell. So these proteins with the little red dots on them, that would be your His tag. And then you would lyse the cell to release all of the proteins. This can be done either under native conditions, where we preserve the structure of your protein, or under denaturing conditions if you're not too worried about the protein structure. A great advantage of this technique is that your tag is linear, so it's not dependent on the structure of the protein, which may be the case for other ligand binding sites, such as the glucose binding protein we just looked at. You would then load the mixture of protein on the column, wash away any unbound proteins or proteins that don't have the tag, and then use free imidazole, which will compete for binding to the column and elute your protein of interest. Now, you can buy small columns that fit into a centrifuge, such as this nickel NTA spin column, and the samples will then be spun down into very concentrated small volumes of elution. And this is great versus waiting for samples to elute by gravity on a regular column. Dialysis is another useful purification experiment that can help remove any unwanted molecules. A dialysis bag or cartridge is a semi-permeable membrane that allows small molecules to pass through. It's kind of like the opposite of size exclusion, where only larger things are kept inside the dialysis tubing. You would then place it in a beaker that doesn't contain any of the molecules you want to get rid of, and the small molecules will travel out through the pores in the tubing based on diffusion. This can remove any high salt that you used maybe for ion exchange, or the small molecule ligands that you eluted your protein from the affinity column with, and you can also change the pH of your buffer in case you want to change its conditions as well as salt concentrations. And this would be known as buffer exchange, where you have one buffer inside the bag and another outside. So for example, if we did use pH on our ion exchange column, we might want to go back to the original pH that the protein prefers. Your protein of interest will then remain in the bag due to its larger size, and then you could remove it and study it under the new conditions. HPLC, or high pressure liquid chromatography, is one way to separate proteins more rapidly and achieve better resolution between the peaks of proteins coming off of a column. So here's another example of an elution profile where instead of measuring volume, we're measuring the time a protein spends on the column, and again we're measuring the absorbance on the y-axis. So when a column separates based on hydrophobicity, this would be known as reverse phase HPLC, where molecules that interact more strongly with the column elute later, such as those in the fifth peak, and proteins in peak 1 may represent ones that aren't very hydrophobic and don't interact with the column. So this technique can be really good for smaller peptides. So on the right, you can see that example, and you can see how there's not much overlap in the peaks if you're trying to purify one protein versus another. In order to see whether or not you've actually purified your protein from a mixture of proteins, you'll usually run a gel where samples are loaded at the top and then separated based on size. In this case, the larger proteins take more time to travel through the gel, and the gel really acts like a sieve, allowing smaller proteins to travel more quickly. In the first lane, this could represent your crude extract or homogenate. So this is what you got when you lysed your cells. And then lane 2 might be the soluble cytoplasmic fraction that you got after centrifugation. You could then run your sample on a series of columns. So first ion exchange, and whatever sticks and is eluted could then be put onto a gel filtration column that fractionates proteins within a certain range, shown here. And then finally, we would have an affinity column that would be specific for your protein of interest. Hopefully by the end, you do have a pure sample of protein and we don't see any other bands. And when you're running a gel like this, you would only be loading a small amount of each of these fractions that you've saved along the way. So you'll still have the rest of your sample for other experiments. The types of gels that we use for proteins are called SDS-PAGE, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. Sodium dodecyl sulfate, or SDS, is a detergent that is used to denature proteins since it has hydrophobic and hydrophilic regions, so it is an amphipathic molecule. And you can see the structure on the top right. This molecule is going to interfere with the non-covalent interactions within a protein structure and will coat the protein with negative charge, solubilizing it into linear chains. These chains will then migrate through the polyacrylamide gel, with smaller proteins migrating faster than larger proteins. Note, this is different to size exclusion or gel filtration columns where the larger proteins will travel faster. Now you could also run a lane of molecular markers on that gel that I just showed you in order to deduce the size of your purified protein. The key take-home messages are that proteins must be purified from other cellular components before we can study their structure and function. Centrifugation is one way we can separate out soluble proteins from other larger insoluble macromolecules and organelles in the cell. And then we can exploit the fact that proteins differ based on their size, charge, affinity, or hydrophobicity in order to use the various types of chromatography. When we want to assess a sample's purity, we can use SDS-PAGE, which will separate proteins based on their mass, and then we can use stains like Coomassie blue to visualize these colorless proteins. We can also use these stains in protein concentration assays, or we can use a protein's absorbance at 280 nanometers, which arises due to the presence of aromatic amino acids. Here's something to think about. How could you purify two proteins that only differ at a single amino acid residue? So for instance, the wild-type hemoglobin has a glutamate at position 6 in the beta chain, while sickle cell hemoglobin has a valine. So if you think about the properties of these amino acids, which technique would be best in order to separate one protein from the other? Don't forget to do the 2 brain break questions posted on Quirkus before watching the next lecture.